I'm going through the stages of meiosis with you. Remember, meiosis has two main purposes. It's to halve the chromosome number, and it's to increase genetic diversity. First comes interphase. Interphase still has the three parts that it had in mitosis. G1, where the cell grows, S phase, where the DNA is replicated, and G2. So the important part of this for cell division is the S phase. So the DNA is already replicated before cell division begins. The first phase of meiosis is prophase 1. In prophase 1, the homologous chromosomes pair up. Remember, you have one chromosome from mom, one chromosome from dad. They synapse, or they come together to form a tetrad. I'm going to show you a diploid cell where the diploid number is 4, because we have 4 different colors. So we're starting off with a diploid number of 4. You can also see that when the homologous chromosomes come together to make a tetrad, you have crossing over. So that's where mom and dad's chromosomes meet, and then they switch genetic information. So part of mom's goes to dad, and part of dad's goes to mom. So in this case, the colors would switch. So the red and the purple would switch places. Same thing happens on the smaller chromosome. The next phase, meiosis 1, or a metaphase 1, the homologous chromosomes line up on the metaphase plate. You have independent assortment in metaphase 1, so that means that not all mom's chromosomes go to one side and dad's chromosomes go to the other side. They line up independently. In anaphase 1, the homologous chromosomes will separate, so they're pulled to opposite sides of the cell. Telophase 1 and cytokinesis. So your nucleus reforms. You have cytokinesis, where the cell membrane starts to pinch off, so you get 2 cells. Notice that in each cell we only have 2 colors, so we have already half the number of chromosomes. It's already a haploid cell, 1N, because we just have 2 colors and not 4 colors. But we still have an X, we still have sister chromatids in each cell, because we still have a duplicated copy of the chromosome. So now we have to switch to duplication in the next phase of cell division. So now we've got prophase 2. Chromosomes condense, prepare for cell division, mitotic spindle starts to form. Metaphase 2, the chromosomes have lined up on the metaphase plate. Notice they're not paired up anymore because you don't have homologous chromosomes. Anaphase 2, the sister chromatids are going to separate. So we're separating those duplicated copies of the chromosomes. Telophase 2 in cytokinesis, the nuclear membrane starts to reform. The cell membrane pinches shut, so you end up with 4 cells. They're all haploid. Remember, this is how you make sperm and egg, where you want half the number of chromosomes, so that when the sperm and the egg come together, you end up with a diploid zygote. So in humans, you have a sperm that has 23 chromosomes and an egg that has 23 chromosomes. So you get a zygote that has 46 chromosomes, so that you don't double the number of chromosomes every generation.